All right, now let's talk about sponges. So sponges are going to be the most primitive animals that you will be responsible for. They basically kind of act as individual cells that are glued together by this like extracellular matrix. Okay, so sponges, remember, so I'll highlight all the names and classifications you'll probably need to remember. So definitely remember the phyla name, Porifera. All right, pores and sponges, I guess, is a good way to remember that. Sponges are kind of pory, porifera. Um, okay, they are all filter feeders, so they basically rely on filtering water through their bodies and collecting all the food bits that float with that water. They're sessile, they don't move around, and they don't have any germ layers. They don't have true germ layers, and they lack symmetry. So the first thing we're going to talk about are some specialized cells that you need to know. There's a couple that are in the slides that are probably most important, but there are a bunch in your lab notebook, so I'll go over a couple of those. Number one, the most important one, probably the canocytes. Canocytes. And so they basically look like this. They have this collar, this netting around them, and then there's a flagella. And this flagella beats and causes water movement through the sponges and really drives the entire feeding of the sponge. So it creates water movement, and then food particles get collected in this collar. They're also called collar cells, and that's what the sponges use to collect their food. They both create the current to move the water through the sponge as well as have the nets to collect the food. So each cell type has got its own specialized function, but I'd say this is one of the more important ones. Okay, the next one in the slides that they want you to know about are the porocytes. So these are going to be the cells that make the pores, or the cells that line the pores of the sponges. And we'll get into the pore structure in a little bit. Just let's go over the cell types first. Next, you'll have lymphocytes and chelinocytes, which make collagen. And that kind of acts as the extracellular matrix that binds all these different cells together. Okay. Another one is the sclerocyte, which makes spicules. Again, we'll talk about what those are in just a little bit. I guess in addition to these lymphocytes and chelinocytes, you also have in the demosponges democytes, which also make a specific kind of collagen in the demosponges, and we'll get to that again in a little bit. Finally, the last cell type that you're responsible for are the amoebocytes, which are basically they are totipotent, which means they can become any of these other cell types. They can become any of these other cell types, but they kind of crawl around through the rest of the sponge material, and they also, in addition to becoming other cell types, they transfer food from the chelinocytes, or sorry, my bad, the canocytes. They collect food from the canocytes and move it around through the sponge to give to these other cells. Okay, so now that we have the cell types down, let's move on to the actual body plan of the sponge. So all sponges as a whole will look like this. You have a big pore on the top, or big opening on the top, called the osculum. And then you have these little pores on the outside of the sponge called ostia. And what happens is water will come in from the outside and go through the ostia, which are the pores, So these ostia basically consist of porocytes. These are the pores that we were just talking about. So they go through these pores and they go into the sponge and then outwards from the osculum. Okay? This is water movement. And what is causing this water movement to go up through, into the sponge and then out through the osculum is the work of the canocytes. Moving water. So we'll take this example of this one simple sponge. So you got these pores, ostia, and then on the inside, for this example of this simple sponge, you have these canocytes that are beating their flagella, and this moves water from the outside inwards and up through the osculum at the top over here. Okay, so let's go through the different body types of these sponges. So we go from asconoid, and these will be important. You'll need to know these names. Ciconoid, and at the bottom we'll do the leuconoid sponges. The asconoid, let's draw out the main body plan first real quick. So these are all the osculum. I'm going to draw the oscula with water coming out. And let's draw the pores. So these are the pores. Leuconoid. Okay. And I'm going to use another color to indicate where the canocytes are. So where the water's, where the source of the water movement is, and also where the sponges are collecting their food. So remember, wherever the canocytes are where the sponges are collecting their food, because they got those collars that filter out the water, the nets, right? And so if you sum up, so let's say these sponges are around the same size from the outside. What you'll notice is that as you go from asconoid to ciconoid to leuconoid, the amount of red is much more in the leuconoid. Why that is there's just all these different small chambers. So the water will come in from the asconoid from the outside through these pores to the inside and then outwards. And so all this red will collect the delicious material in the water. In the ciconoid, the water will come through here, will fork out these multiple tunnels that are lined with canocytes and then into the chamber of the sponge and then outwards through the osculum. The leuconoid, now there's this massive network of different small chambers that are lined with the canocytes. So basically, as you go from asconoid to ciconoid to leuconoid, you get more surface area of red. There's more red, which means there's more efficient filtering of the water. So if you get the same amount of water going through these sponges, there's more opportunity for all the delicious things in that water to be filtered out by the leuconoid because there's more surface area. So you can say, I guess, that the asconoids are the least advanced sponges. The ciconoids are secondly least advanced. Not a great way to say that. The asconoids are the least advanced, ciconoids are medium advanced, and the leuconoids are the most advanced sponge types. And it all has to do with the efficiency of the body plan and how easily and how well they're able to filter out water. Okay, so now let's go on to the different classes of sponges. So you'll also need to know the different classes and their names. So you'll have the calcareous sponges. So class Calcarea, the demo sponges, I don't think they actually make you know the scientific classes, but it might be good to know. Maybe to look out on your own. So the final ones are the hexalinid sponges or the glass sponges. Remember, in lab, there were no glass sponges, so you're not responsible for knowing them necessarily, except a couple of general facts about their biology. What separates them apart? Number one, at least in how we describe them in lab, is their skeletal system. So basically, the support system, all the different things that give them structure relative to each other, right? Links those cells together. So the calcareous sponges, they have spicules that are made out of calcium carbonate. And we'll get into what spicules are in a bit. Demosponges, their spicules, and the glass sponges, both their spicules are made out of silica. And demosponges also have an extra specific kind of collagen, this extracellular matrix that binds all these cells together. It's very soft and pliable, yet firm, so it gives it, you can imagine like those sponges that you use for art class, right? It has like the natural ones. You can push them in and it kind of has a little bit of give, but it'll bounce right back. And that, all that external cellular matrix is made out of spunkin. Remember that terminology. So demosponges, hexalinid sponges have spicules made of silica, and then calcareous sponges have spicules made out of calcium carbonate. Now, spicules are kind of these caltrops-like looking crystalline skeletal, elements that are embedded inside the sponge connective tissue. So you have the sponge connective tissue, and inside all that mass, you have these spicules. They're sharp and they act as reinforcement, but they also act as predator deterrents in these sponges. And again, spicule chemistry is important for delineating these sponges because it's hard to tell sponges apart from their appearance if you're not an expert. And that's really the way that we did it in lab anyways. So remember, glass sponges and demosponges are silica, and then calcareous sponges are calcium carbonate. Cool. The demosponges and the hexalinid sponges, the glass sponges, they are more advanced sponges, quote unquote, and so they only have this leuconoid body type, glass and demo. Whereas the asconoids, the synconoids, and leuconoids, all three of them can be found in class Calcarea. And again, this reflects their evolutionary history. So you have Calcarea branched out from there, and then hexalinids and the demosponges more recently branched out. This is the common ancestor. Okay. Finally, to bring it all back together, how can we tell apart these guys insofar as inside the laboratory? And we did that in one specific way where we treated the sponges with HCl and I forget which strong base, potassium hydroxide. Anyways, so both of these chemicals, strong acid and strong base, are pretty caustic, and they will denature and dissolve a lot of the sponge tissue, right? And what's left over is the skeletal elements, so the skeleton. And one of these skeletal elements are the spicules. Probably the most prominent one that we'll talk about are the spicules. And the spicules, again, can be made out of calcium carbonate or silica. Now, in a strong base, once you dissolve all this fleshy material, you get the spicules. It does not interact with calcium carbonate, nor does it interact with the silica. But acid will dissolve calcium carbonate. So if you have a sponge mass and you dissolve it in HCl and KOH, If this is a demo sponge, you'll get spicules here and some gross denatured and broken down cellular mass, and same in the KOH. But in the calcareous sponge, once you treat it with a strong base, again you'll have spicules and some degraded cellular material. But in the HCl, in the strong acid, there'll be no spicules left. You'll just get degraded glummy material because these spicules got dissolved by the strong acid. Okay? And so that's how you differentiate between these guys in the laboratory. And that should wrap up sponges.